Highly pathogenic avian influenza is a devastating poultry disease that continues to circulate in wild birds and domestic poultry, and has infected and killed people. Experts are concerned that the virus could mutate and lead to a human influenza pandemic. Because of the impact of H5N1 avian influenza on animal health, human health, local economies, and food security, the FAO, OIE, and WHO encourage and support control and eradication of this disease in poultry. This DVD is the fourth in a series on factors to consider in the use of vaccination as an aid for control of H5N1 avian influenza in poultry. The focus of this DVD is avian influenza diagnostic laboratory tests, surveillance programs, and selection and use of vaccines. The information is presented by two avian influenza experts. Mr. Dennis Senni is a senior microbiologist at the U.S. Department of Agriculture's National Veterinary Services Laboratories in Ames, Iowa. He recently led hands-on courses for highly pathogenic avian influenza diagnostics and trained individuals from 79 countries. He has traveled to countries dealing with outbreaks of H5N1 avian influenza to provide diagnostic training and assistance. He will cover the diagnostic infrastructure, surveillance capabilities, and disease reporting systems needed to support an effective H5N1 vaccination program. Dr. David Swain is director of the U.S. Department of Agriculture's Southeast Poultry Research Laboratory. He has conducted research on the avian influenza virus and the use of vaccines to control this disease for over 20 years, and he has published widely on this topic. He serves as a consultant to international organizations, including the FAO, OIE, and WHO, and has assisted many countries in their efforts to control avian influenza. He will discuss how H5N1 vaccines work, the ways they are used, and the different types of H5N1 vaccines available.
The diagnostic laboratory has always played a crucial role in the diagnosis of avian influenza because there are no pathognomonic lesions or clinical signs that will distinguish avian influenza from many other poultry diseases that occurred worldwide. And therefore, the diagnosis of avian influenza requires a laboratory test. And in this presentation, we will first of all discuss some of the goals of surveillance. We'll look briefly at the differences between the goals when there is no disease versus some of the goals of surveillance when we have outbreaks where vaccination is practiced. And then we will also look at some of the appropriate diagnostic assays that could be used, both in terms of detection of the virus or its specific RNA and or its specific antibodies. We'll also look at some of the sampling methods, some of the types of samples that are most useful, and the number of samples that would be useful to give certain levels of confidence that if the disease or virus is present in a flock, that it will be detected by the surveillance methods used. And then we'll discuss briefly about some of the laboratory infrastructure required, a little bit about quality standards, development of laboratory networks that have been shown to be very useful in terms of response, and a little bit on laboratory safety. And then finally, we'll end up with a few comments about reporting of results. Now, obviously, the primary goal of surveillance, both in the absence of disease as well as when vaccination is used, is to have early detections. And this is particularly important when you have situations where a country is considered free of avian influenza. Many countries, including the United States, have learned the hard lesson. And we know now that an early diagnosis and quick response can really limit the economic impact of infections caused by notifiable avian influenza. And when I say notifiable avian influenza, I'm referring to either H5 or H7 subtypes. This is either the detection of the virus by virus isolation or molecular assays, or the detection of specific antibodies to these two subtypes. Surveillance can also help prevent the emergence of highly pathogenic forms of the disease. We have clear evidence now that highly pathogenic avian influenza actually emerge from low pathogenicity viruses that have been allowed to circulate in poultry for a period of time. So by detecting it early and eliminating these low pathogenicity viruses, we hopefully can prevent the emergence of the highly pathogenic forms. And of course, surveillance in the absence of disease will give us the best opportunity to quickly diagnose highly pathogenic avian influenza should it be introduced into a country either unintentionally, such as we've seen in many parts of the world with movement of this virus in wild birds, or perhaps in this climate in the world today, we also have to consider the possible intentional introductions through acts of bioterrorism. So these are all some of the main goals of surveillance when no disease is present. We also need to conduct surveillance to assure that our commercial poultry is free from notifiable avian influenza. And this would be the purposes of trade and or consumer confidence. We have seen in many parts of the world where certain infections of avian influenza have resulted in a significant drop in consumption of poultry products because the consumer cannot distinguish infections reported by low pathogenicity viruses from those that we've seen causing human infections of the highly pathogenic H5N1 in many parts of the world. And of course, to do this, you have to have a combination of both a good active and passive surveillance programs. And doing so, we will allow then good transparency of disease reporting to give some indication of what the status of the national flock is in regard to avian influenza. By having good, reliable, and transparent reporting, we increase the credibility in the international arena, as well as I already mentioned, decrease the fear factor that the public has with the mention of avian influenza these days. And finally, we need to monitor backyard and wild birds for notifiable avian influenza infections with the ultimate goal of preventing these infections in commercial poultry. Now, although some of the goals of surveillance when vaccination used is a little different, still the primary goal, one of the primary goals is to detect infections in even vaccinated populations. And this is either pertaining to the detection of the virus or specific RNA or antibodies. Now we see that when vaccination is used, we have some tests that are no longer very valuable in terms of surveillance. Antibody surveillance is one of these. Unless you can differentiate antibodies that are produced against the vaccine versus infection, antibody surveillance is going to be of very little use. To also, some of the goals of surveillance in vaccinated populations is we need to monitor the effectiveness of that vaccination program. How well are the birds responding to the vaccine? Do we have the correct virus in the vaccine? How many doses do we need to provide adequate protection in these birds? How good a job is the vaccine crews doing? Are they getting 100% of the birds, or are they missing a certain number of birds in each flock? And of course, it's also important to monitor the shedding of these viruses after challenge to make sure that these vaccines are providing adequate protection. If outbreaks are allowed to continue for long periods of time, we know that these viruses are going to change. They're RNA viruses and they're prone to make mistakes, and they will continue to change. So we need to continue to monitor viruses during the course of an outbreak to make sure that we know that our diagnostic tests are still going to be sensitive in detecting these infections. And then also allows us to make good decisions on the proper strain used in vaccines. And probably the most important goal of surveillance is to reduce the likelihood of human infections. I think a lot of the countries that have implemented vaccination to control the high-path H5N1 are countries that have reported the most human infections, and I think this is a really important part of any vaccination strategy. So let's look at some of the diagnostic tests that are available. Now, it doesn't matter what test you decide to ultimately use, it's important to know that the test that you are going to use has been properly validated. And it should be properly validated for its intended purpose, or in other words, it should have fitness for purpose. In other words, serologic tests, such as an AGID test that is shown to be pretty reliable in gallinaceous birds, such as chickens and turkeys, may not work very well in ducks, and it's not known to be a reliable test. So you have to know what is the value of this test and how is it supposed to be used. The same could be said for some of the molecular tests. Tests that have been validated for chickens and turkeys using tracheal or oral pharyngeal swabs may not have the same level of sensitivity in ducks using cloacal swabs. We need to know a little bit about these diagnostic tests. How sensitive are they? What is the limit of detection? And certainly it is important to know the diagnostic sensitivity and specificity. With all the other diseases that are circulating in populations, we don't know what interaction these infections might have with some of the diagnostic tests. So we need to know what the sensitivity and specificity of these tests are. And of course, each test must be shown to have good reproducibility and robustness so that it can be used in many different laboratories with the same results. Now, as far as antigen detection, diagnostic tests, the three that are most commonly used, of course, are virus isolation. There are a number of molecular assays that are being used, conventional RT-PCRs, real-time PCRs, and nucleic acid sequence-based amplification, or NASBA tests. And then we have the antigen capture immunoassays, which provide very quick turnaround time and can be used often on the farm if necessary. So let's look at virus isolation, either using embryonating eggs or cell culture. These have been used for years to diagnose avian influenza. They're used because they're sensitive to all subtypes of influenza. And particularly in the index case, it's very important to make an isolation of that initial virus to allow full characterization of this virus in the laboratory. Know what its biological properties are, some of the molecular characteristics that could help in determining where this virus might have originated. But virus isolation is not for all laboratories. It's very expensive, requires special facilities. It's certainly not specific, because it will support the growth of many other avian pathogens, both viral and bacterial. We may have some low sensitivity to some of the wild bird isolates. Some recent studies done in wild birds comparing PCR-type assays to virus isolation. Usually, in many cases, virus can be isolated from only about 30% of the positive PCR-type samples. So there is some issues as to whether the egg will support the growth of non-poultry adapted flu viruses on a regular basis. And of course, you can have false negatives due to the mishandling of the diagnostic specimens. Now, the molecular detection assays have many advantages. I mean, they're very fast, they're very sensitive. In some cases, we have the ability to differentiate H5 and H7 subtypes very quickly. And we've had very good success of even determining the pathogenicity of H5 and H7s from clinical samples by doing PCR and then sequencing the hemagglutinin cleavage site. The cost of these tests varies. Some are relatively inexpensive, about $8, and some of the NASBA-type tests may reach up to $50. But they have potential for high-throughput testing. In outbreaks, the laboratory sometimes is very much swamped with high numbers of samples, and some of these platforms can use either 96-well or 384-well systems that allows for testing of large numbers of samples. But it's expensive. These usually run $25,000 to $90,000 for startup costs. They are very sensitive, and because of their sensitivity, we have to be very careful with false contamination in the laboratory. And so it requires special facilities, separate rooms, say, for extraction of RNA, a different room for setting up PCR reactions, and then yet a third room for doing post-amplification evaluation of these samples. These tests do not differentiate live from inactivated virus. So if you're doing environmental sampling after depopulation of a flock, for example, to look for live virus, PCR is not going to be the choice for that purpose. Virus isolation will be much better. And of course, there's always the problem of false negatives. We've shown that certain type of diagnostic specimens have PCR inhibitors. We have problems with extracting the RNA from dirty type samples that have a lot of protein present. And of course, we always have to be considered about the genetic diversity of influenza, that our primers and probes simply are not sensitive to detecting a particular strain of virus. Now, the antigen capture immunoassays, there are a number of these that have been developed recently. They're very quick. They can be used on the farm, doesn't require any special facilities. But they're not as sensitive as either the virus isolation or PCR. In work that's been done here in the United States, we've only shown about 70 to 80% sensitivity when compared to virus isolation or PCR. And of course, you have to be concerned about false positives because of sample quality, and usually because vaccination suppresses the amount of virus that's excreted from infected birds, they're probably going to have low sensitivity in vaccinated populations. This diagram, which comes from the OIE Terrestrial Animal Health Code, really outlines the strategy for all three of these methods. Virus isolation on the left, and then in the middle we have in turquoise the molecular approaches, and then on the far right the antigen capture tests. And you can see on the virus isolation, or the far left part of the diagram, virus isolation gives you the capability to do everything that you really need to do. It has complete subtyping of any of the 16 hemagglutinin subtypes. We can determine the pathogenicity in chickens. We can also determine the molecular sequence at the HA cleavage site, which is important in differentiating low from high pathogenicity viruses. The molecular detection assays in the middle will allow differentiating between H5 and H7 viruses, but it really haven't developed the capability to individually subtype some of the other subtypes. But we can, in many cases, determine the virulence of the virus by sequencing the HA. And then finally, on the right side of the antigen capture test, really these are pretty much limited to identification of an isolate as influenza A. There are some H5 specific antigen capture tests available, but the sensitivity of the monoclonal antibody may make it marginally reproducible if the virus changes at all in the field during an outbreak. So if we look at antibody surveillance, antibody surveillance is most useful before and after outbreaks. Before outbreaks, to know if you have any infections circulating in your poultry, and after outbreaks to confirm, if eradication was your goal, to confirm that you are indeed free of disease and have completely eliminated these infections in poultry. However, when vaccination is used, we see limitations on the use of antibody surveillance, and we're pretty much limited to monitoring sentinel birds that are seronegative. They're placed in flocks to determine if there's any circulating virus in these flocks. And also, it's important to determine how well the vaccines are working. What kind of an antibody response are we getting? But unless we can use a DIVA strategy, in other words, differentiate the vaccinated antibody from the naturally infected antibody that's produced, serology is not of a lot of use in these situations. There are two types of antibody detection systems. One is a type-specific test that will detect antibodies to any of the 16 hemagglutinin subtypes. Some examples are Orgagel immunodiffusion and ELISA. And then we have the subtype-specific tests, such as the hemagglutination inhibition and neuraminidase inhibition tests. The type-specific test for the AGID, for example, is very easy. It's inexpensive, requires relatively few reagents, and it has the ability to test multiple species, with the exception of ducks. It doesn't show very good sensitivity for ducks, although there are not very many diagnostic tests that are very useful for looking for antibody in ducks. But it does have disadvantages. It's only semi-quantitative. It will not give you a titer. It's just a positive or negative, or a strong positive or weak positive. And it requires a certain amount of training to make sure that the interpretation in one laboratory is going to be the same as an interpretation in a second laboratory. The ELISA has some advantages over AGID in that it's usually commercially available. And that means that these are probably some standardization in the kit. They're very quick. They can be semi-automated. If you have robotics, you can test quite a large number of samples by ELISA. But it does require special, expensive equipment. And most of these are species specific. And most of them are licensed currently for either chickens or chickens and turkeys. But any positives on either the AGID or ELISA require further confirmation and testing. These type-specific tests really cannot be used or really are not of much value when vaccination using conventional killed vaccines are used, because these tests cannot differentiate vaccine or antibodies that are elicited following vaccination versus those that are elicited after natural infection. But they can be used in situations where recombinant vaccines are used. These are vaccines that express the hemagglutinin antigen, so that the antibodies produced are specific for the H determinant of the virus, and therefore do not interfere with the type-specific tests. The subtype-specific tests, such as HI and NI, are very good, particularly for evaluating the vaccine reactions. Typically, titers of 1 to 32 or higher are indicative of protection, and that birds that have these titers or higher titers probably will resist clinical disease following vaccination. And they're fairly quick and inexpensive. However, you have to have specific reagents for each subtype. So these are not good screening tools, looking for all subtypes of influenza, because requires 16 different hemagglutination inhibition tests. You can have false positives. And one example that I use in this is when H5N2 was first found in Central America, one of the private laboratories there that was doing diagnostic work found some AGID positive flocks. But the only antigen they had in the laboratory was one with an H5N2 subtype. So they thought, well, let's give it a try. So they tested the serum, and they had some low HI titers. So their conclusion was it must be H5N2. In reality, it turned out to be that these serums were positive for H5N2, and that the inhibition was due to the interaction of the neuraminidase components. So if you're going to do HI tests, then the laboratory should also have the capability to do neuraminidase inhibition tests to avoid using antigens where the neuraminidase and in the antigen and in the field serum are the same. But again, these cannot distinguish antibodies from vaccination versus infection. What is really needed is improved DIVA tests. There are some tests that have been developed and used successfully in the field. This was first used in poultry for influenza in Italy. We also used it in the United States following an infection of H7 in some cage layers successfully. Most of these have targeted the neuraminidase component of the virus or the non-structural proteins. So in the case of neuraminidase, what you do is use a vaccine in the field that has a different neuraminidase than the field strain. And then you develop a test that will detect the antibody to the field strain. So if it's positive on this DIVA test, then you know the birds were infected with field virus and not vaccine virus. But we really need to focus on the development of better DIVA tests. We don't have a lot of capacity for large-scale testing. Some of these have been developed for use of ELISA, but still we only have currently tests available for N1 and N2 subtypes. We also need to continue work on the development of marker vaccines with companion diagnostic tests that will allow us to quickly identify those flocks that are vaccinated from those that are field exposed. But again, here's a diagram, again from the OIE Terrestrial Animal Health Code, chapter on avian influenza, outlining the strategies for surveillance in unvaccinated populations versus those in vaccinated populations. Unvaccinated populations, you can use both type-specific antibody tests as well as subtype-specific tests and have full identification of any of the 16 subtypes. However, in vaccinated populations, as you see on the right-hand side, we have two choices, either using inactivated vaccines that have homologous neuraminidase, and you have the options of doing antibody surveillance in the sentinel birds and in possibly DIVA test looking at the non-structural proteins. When you use heterologous neuraminidase vaccines, we have the option of developing DIVAs for the non-structural protein as well as looking for antibodies in sentinel birds and the development of the DIVA-specific neuraminidase antibody test. But however, the recombinant vaccines allows for doing serology both for the nucleoprotein and matrix protein as well as non-structural proteins or the neuraminidase. So it has a lot more options for serologic surveillance with the recombinant type vaccines. This graph just shows you the relative detection or sensitivities of the various approaches to diagnosis, and this would be in an unvaccinated population. In this situation, we see in the bottom there where the virus isolation and real-time RT-PCR are very similar in terms of detecting low levels of virus, and the duration will be somewhere between 1 day and 24 or 25 days or so. The antigen captures requires a lot higher level of virus, and that way the window of opportunity for detecting positives is much narrower. Probably ranges from about day 2 to about day 12. As far as antibody detection, as you see at the top, about the time that we have peak virus titers when we start to see the appearance of IgM. And so about 5 or 6 days post-infection, you will start to see AGID positive birds appear in the flock. About 9 to 10, 11 days post-exposure, we will start to see ELISA positives, and HI positives will probably persist for several months, whereas AGID is probably going to start to diminish after about 30 to 60 days. So after 30 to 60 days, we may start seeing individual birds become negative by the AGID test. If we look at vaccinated populations, we see that we pretty much are limited to either conducting virus isolation or molecular tests. We see a lot lower level of virus produced, so antigen capture is going to be below the sensitivity of detection and probably will not be of much value in detecting infections in vaccinated populations. The other thing I want to mention is that this line of virus detection may actually go below the line of sensitivity. In other words, we may not detect virus every time you sample a vaccinated flock that's been infected with the highly pathogenic virus. So essentially, any vaccinated flock that has been exposed to a highly pathogenic virus and there's virus circulating in that flock probably will be positive for the remainder of its productive life. Likewise, antibody levels, either by AGID, ELISA, or HI, will remain high, but probably of very little use unless there is a DIVA strategy used. So what about surveillance sample collecting? Certainly, the most productive samples in situations where vaccine is used is you want to take samples from areas that are not influenced by antibody, and that would be the oropharyngeal or tracheal swabs and cloacal swabs. You can pool these to reduce the number of samples that actually are tested in the laboratory. We have made successful pools of 5 for many years without any particular problem. Tissues from vaccinated flocks suspected of being infected with high-path AI are of some value, particularly the mortalities. If you have mortalities, brain tissue is very good because there's a lot less antibody in the brain than compared to lung or spleen or some of these other tissues. It's important that you do not pool tissues from different birds because some birds may be antibody positive, which could then neutralize the virus from other birds that may be more acutely infected. The specimens that are of use for antibody detection, of course, is serum, plasma, egg yolk. Egg yolk has some advantage over serum, is that you can collect eggs from processing plants rather than having to go on the farm to obtain these samples. That way you avoid any biosecurity risks of transmitting disease from one farm to another. But any of these 3 type samples can be used in any of the serologic tests that are typically used for flu. But as far as the number of tests, really is dependent on the confidence that you want to have in detecting positives. And I have on this particular page some various sizes of flocks ranging from 50 to 10,000. The confidence intervals either of 95% or 99%, and the left-hand column we have the prevalence of infection within a flock. So you can see that using 95% confidence interval of infection rates of 10% or greater, in a large population of 10,000 birds, we would need to collect 29 samples. In a population of 50, under the same 95-10%, we would need to collect 22 samples. And you can see that the lower the prevalence within a flock, the larger the number of samples you need to collect to assure that at least one of those samples in that sampling will yield a positive result. Now, another way to look at this is that if you don't have this table with you when you're out in the field, you can use the 95% confidence determining the number of samples by using 3 times the interval. So in other words, if we have a prevalence of 1%, the interval is 1 in 100. So 3 times 100 would be 300 samples. In the previous table, I think the number was 294, so it's relatively close. Likewise with the 10% prevalence, the interval will be 1 in 10, so 3 times 10 would be 30. And I think in the table it listed as 29. So you can give a rule of thumb if you're away from a table and need to collect samples. But one thing is important, is that numbers are not what the goal is. The goal is to detect infections of flu. And to do that, we need to use targeted surveillance. We don't just want to go and have random testing of birds. So we need to design monitoring schemes, either weekly or monthly or whatever, of both the commercial, backyard, and duck populations to make sure that we will detect infections if they are present. And we can estimate that within flock prevalence in vaccinated commercial flocks, or any birds, may be very low, and in some cases could be as low as 0.1%. So if we look at that rule of 3 times the interval, if we use the interval as 1 in 1,000, we would technically need to collect 3,000 samples to be assured that we would detect at least 1 positive in that flock. Well, that's typically not very practical, because most laboratories don't have the capacity to do that level of testing. So what we can do is select targeting sampling by focusing on the daily mortalities. Now, if we take this same scenario and assume that the normal daily mortality in a flock is 0.1%, and this can be adjusted depending on the locality, etc. So if the observed mortality rate in a suspect high-path infected flock is 0.2 to 1%, that represents a 2 to 10-fold increase in mortality. So, assuming that the high-path AI virus was the cause of the increase in mortality in this particular flock, we can expect that at least half of these deaths are due to HPAI. Therefore, to detect 50% prevalence, instead of the 0.1, with 95% confidence, we would need to test about 6 samples. It's a lot more acceptable than 3,000. Now, an example of this that we used in an outbreak of low-path influenza in Virginia is we used what we called barrel surveillance. Producers would, on a daily basis or on a specified schedule, would put birds of daily mortality out in these barrels. And then we would have people in the control program visit these premises, collect samples, and send them to the laboratory. And this really was very effective. And we're focusing only on the daily mortalities because that's where you're going to find the virus, particularly in populations that have immunity because of vaccination and or exposure. And this really compensates for a lot of underreporting of clinical disease in many situations. So be sure to target your surveillance. Now, what about waterfowl sampling? This is really not in the scope of vaccination sampling, but I think because of the involvement of wild birds, it's important to make a few notes regarding waterfowl sampling. The sampling of waterfowl, wild waterfowl, may be different than that is necessary and productive for gallinaceous birds. For example, in most cases, infections of influenza in poultry are going to cause primary respiratory infections. And so tracheal or oropharyngeal swabs are very ideal for this type of surveillance. In waterfowl, at least with the low-path viruses and most high-path viruses other than the H5N1, these infections are going to be intestinal primary. So cloacal swabs are going to be the ideal sample. Now, in wild birds, you have to define what your goal of surveillance is. If you're looking for only the high-path H5N1, or are you looking for any influenza that may be present in these wild birds? In the case of high-path H5N1, tracheal or oropharyngeal swabs for mortalities may be all that you need to collect. Studies have shown that since 2002, this virus has become highly pathogenic to wild birds, and so it causes a systemic infection and allows us to focus more on the respiratory tract than typically that we do on the cloacal samples. However, if you want to detect any influenza in these birds, cloacal swabs from both live and dead birds would be preferred. And as far as detecting virus in wild birds, either virus isolation or detection of RNA is the goal. Serology is not going to be a very much help in determining the role of waterfowl in avian influenza. A little bit about the laboratory infrastructure. The laboratory infrastructure will vary tremendously depending on the laboratory's role. What is this laboratory going to do? Is it going to do only strip tests or immunoassays, or is it going to do molecular assays, or is it going to do virus isolation? And the infrastructure required for each of these will be significantly different. But what each laboratory needs to do is define what its role is, what type of testing will be done, and stay within the limits of the capabilities of your laboratory. In other words, if your laboratory is set up to only do minimal work of immunoassays, for example, don't attempt to do virus isolation, because this could put personnel in the laboratory at great risk of exposure and possibly infection. Now, the laboratory also should be involved at some level having a quality program. I think it's important, and every laboratory needs to understand this, is that people have to rely on the test results that come out of laboratories. And if laboratories are not doing the correct things, the reproducibility of the diagnostic test will suffer greatly. So each laboratory needs to have some monitoring of or initiation of a quality control program. This doesn't have to be elaborate. It can be internal, where the director has set forth certain standards for the laboratory, or if you really want to get sophisticated, you can have external bodies such as the ISO organization that certifies laboratories under 17025 if you want a very high level of monitoring. But these laboratories should always use standardized protocols that are in writing with standard operating procedures, and to assure that each of the laboratory people that are performing these tests are properly trained. And of course, as should be done in any case, the proper controls should be available and used on a regular basis within the laboratory. Now, in many countries, the development of laboratory networks has really been beneficial, and this has come about pretty much since 2002. And what the idea of having these networks is to provide a very rapid and sufficient response to animal health emergencies, and this would include foreign diseases or high-path AI. The idea is that you can have results coming out at the local level, but always have oversight from either a reference or a national lab to make sure that there is consistent consistency within this testing and reporting. The idea of the networks is to reduce the response time, to make sure that when we find an infection, we can respond to it, eliminate it quickly so that it doesn't have time to spread. And this provides a tremendous amounts of surge capacity. So I think anybody that's been associated with a laboratory knows that during outbreaks you get enormous numbers of samples submitted to the laboratory, which can quickly overwhelm many of the laboratories. This is just an example of a network that's been developed in the United States since 2002, where each of these labs that you see represented here have been approved to conduct official tests for avian influenza. Each laboratory uses the same protocol and the same technology. Any laboratory that detects an infection of influenza is required to send that sample to the National Veterinary Services Laboratory for confirmation. So here again, we have local testing with some level of federal oversight. By having this network of laboratories, we have the capacity to test more than 25,000 samples a day, which would be pretty good capacity in dealing with most outbreaks. A little bit about laboratory safety. That's very important because certainly people working in the laboratory are at risk for exposure to these various viruses. Many of the guidelines for safe handling of these viruses is found in this BMBL manual, which is the Biosafety and Microbiological and Biomedical Laboratories. This is available on the web. I have provided the website address to you on this particular slide. And what this manual does is define how these viruses should be handled in the laboratory. And this manual defines high-path AI as a BSL-3 organism, which requires certain levels of containment and safe handling practices. Any laboratory working with high-path AI is certainly, since we have had evidence of human infection since 1997 with the Asian H5N1. We also had evidence of quite a number of human infections during the outbreak in the Netherlands. So you have to have the appropriate containment facilities for the level of work that you're going to do. And you use kind of a box within a box within a box concept. Your biological safety cabinet will be the first level of protection. This cabinet is located in a room. This room is located within a building. So you have this box within a box concept. We need to be careful not to create aerosol. So we need to take precautions, such as using aerosol-resistant centrifuge cups to protect any possible exposure if something should break during processing. And of course, we have to be concerned about air handling, sewage treatment, etc., so that we don't endanger the environment or the community where the lab is located. And of course, personal protective equipment should be standardized at a minimum. Gloves should be required, lab coats, safety glasses, and if you're working with high-path AI, certainly an N95 or better quality respirator should be standard equipment. The other issue is shipping of samples. This is becoming very difficult to ship diagnostic samples. And if a laboratory is in a position where if they find a positive, they need to send these samples to a reference laboratory, you need to have these laboratories identified in advance. Know where you're going to send the samples if you have a suspect sample. Be sure to contact these laboratories and make sure that they know that they will be receiving samples in the event that they should have a suspect submission. This is not an easy task. To meet the IATA, or the International Air Transportation Association guidelines, anybody involved in the shipping of materials, including viruses and diagnostic materials, will be required to do a fairly lengthy training session. It involves about 8 hours. So you need to do this in advance so that you don't delay shipping of samples, critical samples, to get a diagnosis. And reporting systems, most of this is just pretty basic. Each laboratory should be able to have a system in place where they can track the sample from the time that it arrives in the laboratory until results are reported. And some of the basic information that needs to be retained in association with each sample, of course, is the owner, the address of that owner, so we can track, provide the results, as well as to get additional samples if necessary, species, specimen identification, and the tests that have been requested to be done. As far as reporting, you need to establish where are the results going. Will they go to a particular official above or whoever manages laboratory? But you want to make sure that the reporting channels are pretty set and don't give out information to people that shouldn't have it. But basically, these reports should contain the methods that were used to obtain the diagnosis. The results should be clearly presented, and you should always have within the laboratory appropriate oversight and validation of the results before they're released to make sure that erroneous results do not leave the laboratory. So basically, in summary, we can say that vaccination certainly affects the selection of diagnostic tests used for surveillance. We know that when vaccination is used, detection of the virus will be more difficult. One of the tools that's taken away is clinical disease. And it's much harder to detect virus at low levels in an immune population than it is when the virus actually produces clinical disease, because you know which flocks then to test. Targeted surveillance will help to reduce the number of samples that you need to test to detect infections in some of these flocks. And of course, the laboratories need to develop sound quality programs to increase the confidence of laboratory results. If people do not have confidence in the laboratory results, then it's going to make control very, very difficult from a field perspective as well. We have to take into account safety as a very important laboratory activity, and of course no job is done until the paperwork is completed. So the report that goes to the field or to up the chain of command needs to be done as quickly as possible so decisions and reactions can be implemented.
In summary, diagnostic support is critical for avian influenza control and eradication. For a successful response, tests must be conducted according to international standards and be properly interpreted. For more information on the diagnostic tests, please see the set of DVDs entitled USDA Highly Pathogenic Avian Influenza Diagnostic Training Course. These DVDs demonstrate the laboratory tests and provide standard protocols for each of the tests. This information is available on the Internet at www.cfsph.iastate.edu/HPAI.
So some of the basic questions about vaccination is, how does avian influenza vaccines work? First, how do they produce protection? And really there are three types of immunity that are involved in this protection process. One, and the most widely recognized, is humoral immunity, but also cellular immunity and the innate cellular immunity are involved in this process. By far the best information we have available shows that antibodies against the hemagglutinin protein are the most protective, although antibodies against the neuraminidase protein are also protective, but somewhat less effectively. And these antibodies are protective against that specific hemagglutinin or neuraminidase subtype. We know from many experimental as well as field studies that the protection afforded is very type specific. Therefore, there is no single universal vaccine that can be used against all avian influenza viruses. That is to say that historically, in the field as well as experimentally, the protection is based upon protection against a specific hemagglutinin subtype. Therefore, vaccines are used and developed based upon which hemagglutinin subtype is the most likely for exposure. Because avian influenza virus infections involve initiation of virus growth in the respiratory and the alimentary tracts, thus the concept of a sterilizing immunity is not really achievable in the field. Just for example, you can look at other respiratory viruses, not only in poultry but other animal species, and see that generally those vaccines reduce the virus growth, but they are not preventative completely of virus replication, which emphasizes the need for biosecurity to prevent cross-contamination and movement of viruses. Therefore, biosecurity enhancements are necessary anytime you use vaccination as a control program. But there are some cautionary points we must discuss and understand. Vaccination is only that single tool in an avian influenza control program for very specific reasons. One is that the vaccination process itself, the usage of a vaccine, can interfere with both the active and passive surveillance programs unless that vaccination and surveillance program are co-developed and properly designed and implemented. Second, the vaccination, if used in the field, can hide clinical signs of avian influenza infection. For example, highly pathogenic avian influenza, which would normally cause high mortality in chickens. In vaccinated flocks, you can have infection, but without the high mortality. Therefore, this would eliminate clinical signs as a primary screening tool to identify presumptive highly pathogenic avian influenza cases for further diagnostic evaluation. And finally, vaccination can impact international trade if it's not done properly. And therefore, any time a vaccination program is under development, it should be done according to those guidelines developed and published by OIE and FAO to assure that the surveillance will adequately detect infection in vaccinated flocks. To emphasize, avian influenza vaccination must always be done properly in the proper context of the risk as well as the proper context of how the application is in the field. But before you vaccinate, you must plan ahead. You must analyze the situation, determine what is the avian influenza status of that country, zone, or compartment. For example, is the infection endemic? If it's an endemic, there will be a continual source of avian influenza virus, and therefore the need for vaccination may be greater than, for example, if it's an active outbreak where you have focal source of avian influenza that you can pinpoint and eradicate by only stamping out programs. Finally, if you have no avian influenza virus present in that country, zone, or compartment, then you have to look at it as a preventative measure and determine what's the risk of introduction and how effective will vaccination be as a preventative. For example, if it's in high-risk areas, such as in poultry outbreaks adjacent to a border in a country where there is a porous movement of animals, then it might be of value to consider developing a preventative program. Or if it's in countries where you have wild bird infections, then it may be of value to reduce the risk by vaccinating certain poultry species. However, in the cases of where there's low risk, where there are no cases in poultry within that particular company or country or within their neighbors' countries, then there may be no need to do vaccination as a preventative. And those risks have to be weighed and analyzed. Also, how will vaccination impact diagnostics and surveillance? In this case, vaccination will eliminate the detection of high mortality as a signalment for a diagnostic investigation. Therefore, you must develop additional strategies using the DIVA concept, differentiating infected from vaccinated animals, to be able to detect infection in those vaccinated populations. In this case, these DIVA strategies, they could use serological strategies or they could use virus detection strategies through a variety of means. And how can this vaccination program be done properly? Well, it has to involve correct vaccines, both in the quality and in the type of vaccines. They have to be properly transported and properly stored. And then finally, they have to be administered properly by a correct vaccination technique. How can avian influenza vaccination be used correctly? For example, one way is vaccination can be used as a tool during an eradication program. Generally, there are two concepts that are discussed about using in vaccination programs. One is a ring vaccination program, which is a general concept used with other foreign animal diseases. And that is vaccinating in a surveillance or buffer zone outside of the quarantine or the outbreak or infected zone or compartment. This actually is quite difficult to use as a control measure with avian influenza and some other diseases unless you can for sure develop a strong quarantine zone so that you do not have birds or animals moving across that buffers or surveillance zone out to an outer side area. To practical uses, a much better way to use vaccination as part of an eradication program is to use it in a suppressor vaccination concept. And this could be done one of two ways. For example, it could be done in the face of an outbreak in birds within that infected zone to reduce the spread and reduce the clinical disease and reduce contamination. The second way is in eradication zones or infected zones after there has been a thorough eradication done. Birds going back into that zone for repopulation could be vaccinated to increase their resistance to potential outbreaks if they're exposed to residual virus. Secondly, vaccination can be used as a tool in management because the vaccination can decrease the clinical disease and reduce economic losses. Much of the vaccine that's being used around the world for H5N2 high-path AI is being used as a management tool to reduce the amount of virus replication and shedding into the environment as a prelude to eventual eradication down the road. This is generally a type of strategy used where the virus has become endemic in domesticated poultry populations and where the need for long-term control by reducing virus shedding in the environment is necessary. Third, vaccination can be used as a preventative tool. Generally, it's used in areas where there's high risk of introduction of avian influenza. For example, it could be in adjacent countries that have a high-path outbreak and other countries that have porous borders. In this case, you would be protecting poultry populations in the free country if there is movement across that porous border. A second place that it could be examined is areas with detection of highly pathogenic avian influenza in wild birds. And you have poultry that are raised outdoor with potential exposure to infected wild birds. In this case, vaccination could be considered and examined as a possible way to reduce the risk. But even if you do decide to use vaccine, it has to be prioritized to what the risk would be. And there should be a priority level determined as part of the goal of vaccination of where and when to use that vaccine. For example, high-risk populations of birds would be the first that should be vaccinated. In this case, in an outbreak zone, either as a ring or suppressor vaccination should be considered. If the country has a large village poultry sector that has infection, in this case, vaccination may be of benefit in reducing susceptibility to other birds within that population. It also may be helpful in outdoor commercially reared waterfowl in reducing their potential to become infected because of the risk of exposure. A second priority, which would be a little bit lower than the high-risk population, are populations of commercial birds that have a high value, such as genetic stocks of pure lines or grandparent stocks. Because of the high individual value of these birds and the difficulty to replace, replicate them, vaccination may be useful in situations where the risk is high. Thirdly, rare captive birds which may be endangered or threatened species in zoologic collections that may have exposure would be a third priority. A fourth priority may be long-lived domesticated poultry, such as egg-laying birds or parent breeders who have a long opportunity for potential exposure. And at the bottom of the list for vaccination priority would be commercially reared birds that are raised indoors because they have a short life and have a lower exposure potential. So when designing a vaccination program, you have to very closely look at a priority development based upon the risk for individual populations of birds in that country, zone, or compartment. There are various categories of vaccine technologies that can be used and have been used. Some are experimental, some have been licensed and used in the field. By far the most commonly used technology is inactivated whole avian influenza virus vaccines. Also, there are in vitro expressed hemagglutinin protein vaccines that have been experimental to this point. Those that are grown in eukaryotic cell culture systems, either plant or animal cells, or in plants or yeast or bacteria or viruses. For example, a baculovirus that has an influenza hemagglutinin gene insert. The other way is to have in vivo expression of the hemagglutinin protein. This is done primarily by using virus and bacterial vectors. And a variety of vectors have been used experimentally, although of these list of vectors, only 2 have been used commercially, the fowlpox vectored system with influenza hemagglutinin gene insert, and the Newcastle disease vectored system with a hemagglutinin gene insert from an influenza virus. And finally, the other category is naked nucleic acid vaccines. At this point, there has not been any commercialization, mainly because of the cost of production and usage of those are prohibitive at this time. So vaccines that should be used in the field should only be those that are licensed. And there are several criteria necessary in examining if that vaccine is sufficiently good enough to be used in the field. For example, in the licensing process, it should be determined if that vaccine is pure. That is, it's not adulterated and contains only the desired compounds and are consistent in their composition to produce a strong, consistent immune response in vaccinated birds. That vaccine should be demonstrated to be safe so that when administered, it does not produce deleterious effect to the vaccinated host or to the environment that they're in. Third, it should be efficacious. There should be studies done to show that it meets minimum quantifiable protection criteria that shows the vaccine is of value. And finally, the vaccine should be potent. That is, it should have sufficient antigen content or a sufficient infectious titer that it will be efficacious when used in the field under a variety of conditions. Now, for example, there are a variety of licensed H5 avian influenza vaccines that have been used around the world. Most of the use has been in two locations. In Central America, over the past 10 years, against H5N2 low pathogenicity influenza. But also there has been a large number of vaccines used in Asia with the current H5N1 outbreaks. The majority of the vaccines used have been inactivated vaccine strains emulsified in mineral oil adjuvants. There's a variety of vaccine viruses that have been used. Most of them are low pathogenic field outbreak viruses of the H5 subtype that have been used. But also there have been the use of infectious clone system to generate, through reverse genetics, a couple of vaccine viruses that go to very high titer in the embryonated egg culture system. And we have examples of at least one virus or a series of viruses that have been used that are highly pathogenic viruses produced under high containment conditions. Also, there have been recombinant fowlpox viruses licensed. There are two of them that have different hemagglutinin gene inserts. One of them, the hemagglutinin gene alone, from Turkey, Ireland, 83, and the second one containing both the hemagglutinin and the neuraminidase genes from A. goose, Guangdong, 96. And the final category that has been used in the field is the recombinant Newcastle virus vaccines, of which there are two available. One is based upon a insert from the H5 gene of A. goose, Guangdong, 96, and the other one from a Mexican H5 virus insert. In all these situations, the hemagglutinin gene cleavage site has been altered from a highly pathogenic cleavage site to that of a low pathogenic virus. When we look at licensed avian influenza vaccines, by far the most of the vaccine that is produced and used in the world uses an inactivated whole avian influenza virus. This technology is a very old technology used for over the past 50 years. It's a very mature technology, usually based upon low pathogenic avian influenza virus field strains with the same hemagglutinin subtype to match the field virus wishing to protect against. These viruses are grown in embryonated chicken eggs in a biosafety level 2 manufacturing facility, and then they're chemically inactivated by one of three chemical inactivants: formalin, beta-propyllactone, or binary ethylenamine. Following inactivation, they're then emulsified in a proprietary oil adjuvant system, and then the product is licensed and then used. Occasionally, highly pathogenic avian influenza virus strains have been used for vaccines, but for their use, the manufacturing must be done under high biocontainment in BSL 3+ facilities. Recently, new technology has been developed which allows the development in the laboratory of more optimal vaccine viruses through the use of reverse genetics, i.e. an infectious clone system. In this system, they use the hemagglutinin and neuraminidase genes from circulating field viruses, but usually alter the hemagglutinin protein such that the cleavage site will be made into a low pathogenic virus from a highly pathogenic predecessor. Then the internal part of the influenza virus is composed of 6 gene segments derived from a vaccine strain that grows to a very high titer. Combining that with the 2 field strain hemagglutinin and neuraminidase genes, they produce a low pathogenic influenza virus that grows to a very high titer in the embryo chicken system, thus producing a high antigen mass. By using this reverse genetic system, the advantage is it gives you a better genetic match with your outbreak viruses. Therefore, you should produce much better immunity that will last a longer period of time. There are many positive aspects of using inactivated whole avian influenza virus vaccines, including it's a mature technology with very low unit cost. It's a well-used technology, very familiar by poultry vaccine manufacturers, and there's widespread understanding of proper administration in the field. And these vaccines give good protection. But there are limitations to what these vaccines can do, and those need to be understood in designing programs where they can be used properly. For example, they do require expensive, labor-intensive parental administration, which only is economically viable in economies where the labor costs are sufficiently low. The second thing is the protective response is very dependent upon the antigen quantity in the dose and the adjuvant system. Therefore, the quality of the vaccine and administration is very, very important. And finally, there is difficulty in identifying infected birds by serological tests in vaccinated populations unless the vaccine and the surveillance program are designed ahead of time to detect the field virus and the vaccine not interfere with that surveillance. The second category are the recombinant fowlpox vaccines. In this case, there are 2 products available, one that has an insert of the Turkey, Ireland 1983 H5 hemagglutinin gene, and the second one has the hemagglutinin and neuraminidase genes inserted from A. goose, Guangdong. There's some advantages of this type of vaccine. One is it can be administered in the hatchery, where you have advantages of high biosecurity in commercial production companies, and there's high quality control of that vaccine administration process. The other thing is it lends itself more easily to the DIVA strategy by using existing antibodies in hemagglutinin inhibition tests to tell you the vaccine has produced an immune response, and you can also do surveillance with existing techniques that measure antibodies against the nuclear protein, the agar gel or the ELISA test. Therefore, if they're negative, indicates no field virus exposure. If they're positive, you have exposure to a field virus. But there are limitations of how this technology can be used in the field. For example, this vaccine can only be used in chickens and not other species of birds. The birds cannot have had prior immunization with a fowlpox vaccine or prior exposure to the fowlpox vector, or it will inhibit the vaccine from growing properly and producing a good immune response. And finally, this vaccine also requires parenteral administration, so you have the cost of the administration and the labor of its use. The Newcastle recombinant vaccines have some advantages. They can be given by aerosol spray or in the eye drop, which means you can do mass immunization and lower the cost of the immunization process. The vaccine also protects against both avian influenza, for which the gene insert is in, either H5 or H7, depending on which gene was put in, and it can provide protection against Newcastle disease virus, which is very common around much of the developing world. This technology can use the existing serological test as a DIVA for avian influenza as well as Newcastle, but there are certain limitations. For example, you have to know the present antibody status of the birds you are immunizing, because if you have high maternal or active immunity, this vectored vaccine will not grow properly and produce a primary immune response. The vaccines can be obtained from a variety of sources. By far the most common source are commercial vaccines, but generally these inactivated vaccine supplies are limited. Generally there are several months lag to produce in emanating eggs, inactivate and emulsify, and get to the field for use. Most of the companies maintain minimal stocks. They usually manufacture and produce those for a very limited market. But if you look at some of the other technologies, for example, fowlpox, recombinant vectored, it's a tissue culture produced vaccine, so it has a much quicker production time, and you can get it within several weeks. The other source of vaccines are through vaccine banks that are developed by various countries, or at least regions of countries, that can be available for emergency use. Historically, these have been kill vaccines where the vaccine virus has been grown in emanated eggs, and the antigen produced, killed, and frozen in storage. Then if a need for the vaccine arises, the antigen is thawed, emulsified, and then administered, distributed in the field for administration. Generally, there's a lag of 1 to 2 weeks of time between when the antigen is thawed out to when it can be bottled and shipped and available. There are several very important field application principles that have to be understood. One is the vaccine is very important. In this case, the hemagglutinin content of killed vaccine, the amount of antigen in each dose, is very important, or the infectious titer of a live virus vaccine. In the inactivated vaccine, the oil emulsion system is very critical to produce a very strong and long immune response. There must be good demonstrated quality control by the vaccine manufacturer, so there's consistent quality of the vaccine, and when immunized between different batches, there'll be a consistent immunity among birds. It should always be recommended to use the full manufacturer's dose and not to cut or reduce the dose that is given to each bird. Proper storage and delivery control must be done. This temperature control is necessary to prevent degradation of the vaccine, which would be an inferior vaccine for use in the field. And then there must be data to show that the vaccine provides protection against the candidate field avian influenza virus. Administration is the primary goal, and the goal is not the actual injection of the vaccine, but the goal really is immunity. So therefore, the skill of the vaccinators is very crucial. They must provide the high-quality vaccine consistently in the correct anatomical site in order to produce a protective immune response. In the vaccinated population, there must be an adequate number of birds immunized to decrease the potential for an infectious cycle to be originated. Generally, the goal is a 90 to 95% immunity within that farm or that zone or that compartment. In this case, we need both high levels of immunity in individual birds as well as population immunity. And the last thing on the administration is that the administration of the vaccine or the vaccination program cannot be viewed as a single administration program. If vaccination is undertaken, it must be realized that multiple vaccinations must be done when naive birds are added to the population. But there must be developed an exit strategy of how the vaccine will be administered, the types of birds, and the length of time. In administration, you must know the species of bird that the vaccine will be used in and the length of life for those individual production birds. This will impact the number of doses of vaccine that each bird will need in order to produce protective immunity. For example, in ducks and geese, there's a minimum of 2 doses to produce sufficient immunity to last for their lifetime. In turkeys, it may take 2 or more doses in adult turkeys to provide protection. Chicken layers and breeders, because of their long life, may need 2 doses. But if you look at meat chickens, generally 1 dose will be adequate unless there are circumstances suggesting that there is excess exposure. In all situations, biosecurity must be practiced because the vaccination crews can also spread virus from farm to farm. Therefore, high-level biosecurity, decontamination of vaccination equipment, decontamination of clothes and shoes and vehicles must be practiced when leaving a farm after vaccination or before entry upon another farm. Environmental factors do impact the success of vaccination. For example, we must know the immunologic competence of the birds. Control must be made for viruses, bacterial, or other conditions that cause immunosuppression, such as infectious bursal disease virus, or chicken anemia virus in chickens, or hemorrhagic enteritis in turkeys. We must know the virus load in the environment. If there is high environmental load of the virus, it may require additional immunizations to keep the immunity at a very high level and provide protection. If we have high-path AI outbreaks within vaccinated flocks, this may tell us that the number of immunizations is insufficient or the titer of immunity is too low, and therefore more doses may need to be used and boosted more often. And finally, because these are viruses that may be around in the field circulating for long periods of time, there should be periodic testing of emerging field viruses against the existing vaccines, generally every 2 or 3 years, to assure that the vaccine is still protective against the field virus. If new viruses emerge that are no longer protected by the existing vaccines, the vaccine strains need to be changed to provide protection. There must be ongoing assessment of the field vaccination program to determine both the immunization success, which is usually done by serologic titers, most frequently by hemagglutination inhibition titers, to show that there is sufficient antibody being produced in the birds and providing long enough titers that they protected for their production life. Or there must be protection studies done that allow challenge of vaccinated birds against the field virus and show demonstration of protection. There also must be continual evaluation of the assessment of the vaccine success by looking for evidence of infection, either serologically using a DIVA strategy or virologically by a variety of methods. And finally, in this vaccination strategy, there needs to be a concept of a quantifiable either zone, compartment, to determine if the vaccination population is being contained or you have introduction of non-vaccinated animals. The conclusions for this presentation on goals of vaccination: vaccination should be used as a single tool in a comprehensive control program. Humoral immunity to the hemagglutinin protein is the primary protective response, and it is specific for an individual hemagglutinin subtype. There are 4 different vaccine technologies used in producing experimental as well as field avian influenza vaccines. They are inactivated avian influenza virus vaccines, vaccines that are produced by in vitro expression of the hemagglutinin protein, either in a tissue culture system or potentially in embryonated eggs, in vivo expressed hemagglutinin protein, usually by vectored bacteria or viruses, and DNA or naked DNA vaccines. Ideally, field avian influenza vaccines should be licensed, and thus they must have demonstrated purity, safety, efficacy, and potency. Always plan ahead. Develop the avian influenza control program before the outbreak begins, and that should include determination of when and how to do a vaccination program. There are very specific goals of vaccination program against avian influenza. It increases resistance to infection. It prevents disease and death. It reduces virus replication and shedding from the respiratory and alimentary system, and therefore decreases environmental contamination by the virus. This reduces transmission to birds and prevents human infections. Decisions to vaccinate for avian influenza should be based on evaluation of the needs and the development of a proper plan for vaccination.
The decision to use vaccination in the control and eradication of avian influenza HPAI is not simple. This presentation provided important information about avian influenza vaccines, how they should be used, and some of the risks associated with their use. For more information on vaccine selection and antigenic drift, please see Dr. David Suarez's presentation at www.cfsph.iastate.edu/HPAI. This DVD on avian influenza diagnostics and vaccines is one in a series of 4 DVDs created to assist government officials and veterinary officers with the decision of whether or not to use vaccination as a tool to control and eradicate H5N1 highly pathogenic avian influenza. In addition to the 4 DVDs, there are 16 individual presentations by avian influenza experts on various aspects of diagnosis, surveillance, response, and recovery, as well as a roundtable discussion on H5N1 issues. Other helpful resources available include a set of DVDs with demonstrations and standard operating procedures for avian influenza diagnostic tests and an avian influenza vaccine database. All of these resources can be found at www.cfsph.iastate.edu/HPAI.